The non-invasive diagnosis of bladder cancer remains a research priority in order to reduce patient burden and healthcare costs. However, despite intensive research for over a decade, cystoscopy remains the mainstay of the diagnosis of new cases of disease and the surveillance of patients already diagnosed with non-muscle invasive bladder cancer. However, in recent years, DNA-based assays have come to the fore and show genuine promise in this setting. In a previous publication, we devised a simple strategy to identify 7 commonly mutated genes in bladder cancer and utilising multiplex PCR to amplify the regions of interest and targeted deep sequencing to identify the mutations. Previous research has also identified the potential advantages of utilising cell-free DNA found in the urine supernatant compared to the cell pellet DNA extracted after centrifugation. With funding from Cancer Research UK, we have been able to expand this work in order to improve the sensitivity of the assay. And so our objectives for this piece of work were to produce a urine test that could identify the majority of bladder cancers with the least amount of sequencing. In the first instance, and utilising our existing collection of snap-frozen bladder tumours, we iteratively and experimentally added genes to the panel, informed by our own sequencing data and publicly available data from resources such as the TCGA. And so we arrived at a 23 gene panel to identify 451 unique mutations which we considered could identify the majority of bladder cancers.
Okay, so when we used the multiplex PCR to sequence mutation hotspots and mutated regions of the 23 genes in 956 bladder tumours, we detected mutations in 96% of the tumours, and that was across all stages and grades of disease. We then went to look at whether mutations in any of these genes were prognostic, finding that several of them are, before moving on to analysing urinary DNA. So we applied the test to DNA extracted from urine pellets, and we were able to detect mutations in 76% of cases, i.e. 76% sensitivity. When we looked at the cell-free DNAs from the urine of these patients, the sensitivity crept up to 84%. So to see why we were getting 96% sensitivity in the tissue, but only 76 to 84 in urinary DNA, we tried a new way of doing the assay. It was a capture-based method incorporating unique molecular identifiers, which allows us to detect very low levels of tumour DNA. Using this method, 3/4 of the false negative urine samples did have mutations detected.
And so we feel that we have described some of the key components of a potential non-invasive diagnostic test for bladder cancer utilising a 23 gene panel. This panel also demonstrates utility for risk stratification and possibly even response to targeted therapeutics. These mutations can be detected in both cell pellet DNA and cell-free DNA, and the key aspect of any future test will be the ability to detect very low mutant allele frequencies in both urinary DNA substrates. Thank you.